Okay, the last topic in or from the prokaryotic gene expression is actually on recombinant gene expression. So this is how we would basically look at how it could be regulated and controlled. But generally, this topic also applicable to eukaryote as well, especially towards the last part where we will see on the gene editing part. So I'm focusing more on prokaryote, okay, because usually prokaryote, okay, is being used, okay, a lot as the expression system, okay, which is for efficient gene expression. But again, both of them actually takes into account. So let's see, okay, how it goes. So basically, it starts off with your recombinant DNA technology. So we both or all of us know, okay, what recombinant DNA technology is. So it basically starts out with your gene of interest, okay, whereby you isolate it, okay, and then okay, you basically insert it into a plasmid, okay, or you have another plasmid that is by your choice of interest, okay, you just cut it out, okay, and then you ligate it and then you just transform it. So once you transform it, okay, you can basically just clone it, okay, into a host, okay, and then later on, okay, you can just purify the clone copies and use it for downstream purposes, for any other purposes that you have in mind. So basically, all this recombinant DNA technology or gene expression starts with the choice of vectors. So vectors is actually very important, okay, it's actually crucial, okay, for your selection, okay. So vectors are DNA with an independent origin of replication. So the keyword here is the ORI, and some selectable or differential markers. So I'm using OR, okay, because a selectable marker permits a host cell to survive in otherwise lethal environment, which means that usually it just survive at its nice environment that it could adapt to, okay, but a selectable marker would allow it, okay, to survive in something or an environment, okay, which is it's not used to. So a differential or non-selectable marker would permit identification of a bacterium by its appearance, okay, which is by its phenotype, okay, kind of like you have GFP, okay, that is basically a differential marker. So vectors could be in the form of plasmids, a lot of it in the form of plasmids. Some of them are also in the form of viruses and viral derivatives. So this is basically mostly true for the use of animal cells because viral vectors, okay, would work much better for animal cells. And apart from that, we also have artificial chromosomes. So all forms of cloning are basically just technical variations on the plasmid cloning. It depends on the type of vectors that you use, whether it's from plasmid or viruses or artificial chromosome. Now, the second element that is crucial in recombinant DNA technology is the host. So, very much like what you know of the general meaning of the host, okay, a host in recombinant technology is the one that housed the recombined DNA. So, it could be either prokaryotic or eukaryotic, and its function is that it must take up the recombined DNA. So the recombinant DNA, okay, basically, okay, has to be made or replicated inside the host. So there are technical approaches to introduce the DNA into the bacterial cells, okay, usually by either transformation, whereby, okay, DNA is added directly, okay, or using transfection. So don't be confused. Transfection and transfection, they actually have similar meanings. It's just that, okay, when general transformation or uptake, or basically how basically it is added, the technical approach, okay, or the procedure is called transformation. But when you use animal cell, okay, to take up or basically to be ligated, okay, into the vector, it is called as transfection, okay, because usually it is the viral vectors that is being used, and okay, basically it is done usually and only in animal cell. So the other approach or protocol procedure that we use is electroporation, and this is done by electric, basically current, okay, as you can see over here, okay. So this is just a diametric diagram. So these two, okay, are done using chemically or chemicals, okay, and this is done via, well, some chemicals as well, okay, but it is also done basically using a current, okay. So regardless of the technical approach, okay, that you choose, okay, it must support the growth of the recombined DNA, because without which then your recombinant DNA won't grow and you wouldn't get your recombinant DNA. So, what to do with the clone or recombinant DNA? So, one, it is actually inside the host. The host can be grown and the plasmid could be easily isolated, hopefully, if nothing goes wrong. But, okay, you have to bear in mind, okay, after you have cloned it, okay, what you have, okay, would be, okay, a mix of positive and negative recombinant, okay, or sometimes what we call as a false positive recombinant. So you need to verify it, you need to select it, you need to screen it, and then you need to sequence it, because this is the ultimate verification. So this could be done directly on purified recombinant DNA, and then later on, once you have sequenced it, you have verified it, you can just express it, okay, to see whether it does or it do really have the similar expression, okay, of the gene that you desire. And furthermore, you can also get a protein from it, okay, and it could basically be made in abundance. Apart from that, what you can do, okay, is that you can do some mutation. You can mutate it, okay. So a lot of mutation is done via site-directed mutagenesis, which is always possible because once a sequence is known, so you could actually alter anything, okay, especially the nucleotide, okay, by your own design, okay. If you know how to design, if you know what you are doing, you could do site-directed mutagenesis. Apart from that, okay, the mutagenesis would also allow new proteins to be designed, and the control elements of the inserted DNA may also be studied and altered, okay, to suit a certain application, or if you want to play around with the gene or to see whether it could function better or lesser and so forth. Now, how do you express the clone? So it's actually sometimes it could be easy, sometimes it could not be that easy. But once the DNA is isolated, so the insert, okay, usually is removed from the vector, okay, because sometimes the vector may play around or may kind of like have an influence on how the gene could be expressed. So it will later be cloned into a vector that contain control sequences for RNA polymerase. So this is what we normally do. This is, I'm taking an example of insulin, okay, over here. So the control sequence, okay, usually contain like promoter in insulin case, and the transcription of the insulin gene, okay, could be increased with the addition of IPTG. So the bacteria, okay, is then transformed, okay, with a recombinant vector, and insulin protein was synthesized from the transcript polymerase by the RNA polymerase. So apart from that, well, it may seem simple, okay, theoretically it's simple, okay, in real life it could take months up until years, okay. So there are problems, okay, with expressing eukaryotic genes in bacteria. Okay? So this is where you can see that this topic is actually a combination of both. So again, okay, taking the example of insulin, because I think everyone could relate to it, right? Okay, you guys know what insulin do to our body, right? So insulin usually could not be properly processed in bacteria, okay, because obviously, okay, it is basically generated, okay, from human animal. So it is also because of the double A plus B chain, okay, in the structure, preproinsulin A, preproinsulin B. So the signal peptide and the center peptide could not be removed by the bacterium. Hence, it is also quite difficult, okay, to be purified, okay, because the signal peptide would cause aggregation of the protein. So when the protein aggregate or when it clumps, okay, purification usually could not be done easily. Okay, what you get is actually very small amount of protein, and purification level is actually not that high anyway. Alright, so another approach, okay, to overcome that, okay, and this is again taking the example of the insulin, is for both A and B chains, okay, to be represented individually. Okay, so this is what has been done, okay, whereby both of the A chain and the B chain, they basically are recombined, okay, separately. Then, okay, only they are combined as a fusion protein. So this was done, okay, to remove the signal peptide from the clone prior to the expression, so that it doesn't play havoc. So when it is cloned, okay, as a fusion protein, okay, usually, okay, there is an addition of beta-galactosidase at its site. So because, okay, the gene is now lacking with its signal peptide, okay, but, okay, at the end terminal, okay, of the methionine, it is replaced, okay, with the 3' end of the beta-galactosidase gene. So this meant that the expression of the clone fusion gene, okay, would produce a protein with beta-galactosidase, okay, on its amino acid terminal end, okay, and proinsulin, okay, at the carboxyl terminal end, C terminal end. So this would help with the purification because beta-galactosidase would not aggregate. So you could purify your insulin much better. And apart from that, okay, you also have to bear in mind, okay, later on, okay, the beta-galactosidase has to be cut off from the insulin, because then your insulin could not be functioned as it should be. So it should not have beta-galactosidase, because beta-galactosidase is not needed, and sometimes it could also play havoc with the expression of insulin. Now, expression of the proteins, okay, using recombinant DNA technology is actually not that uncommon. It's actually quite common nowadays, and a lot of people, a lot of researchers do it. Okay, a lot of countries, okay, and a lot of production of products all over the world. So cloned or amplified DNA can be purified and sequenced. We know that, okay? And it could be used to produce RNA and furthermore protein, or you could also introduce into organisms with the goal of changing their phenotype, okay? Transgenic animal or transgenic plant or anything transgenic. So one of the reasons why recombinant DNA technology usually has such a huge impact on the biochemistry is because it could, so to say, okay, that's why I would put there, okay, in red letters, okay, overcome many of the difficulties inherent in purifying low abundance protein and determining their amino acid sequences. But okay, you also have to be careful because there are some other elements, okay, that needs to be controlled, and you basically need to know a lot about your gene and protein, okay, before you can proceed with this procedure. So the expression of proteins, okay, using recombinant DNA technology usually, okay, would allow the protein to be purified without further characterization. Because we use the fusion protein just now, we know for sure that the protein would be made. So purification could begin with overproduction of the protein in a cell containing the expression vector. And the expression vector we are talking about here could be either prokaryotic or eukaryotic expression vector. So expression vector is different from your typical normal vector, okay, because when we talk about, or when we say expression vector, which means that it helps to express the gene further and better. Okay, so let's have a look at prokaryotic expression vectors first before we look at the eukaryotic expression vector. So expression vectors for bacterial hosts usually are plasmids, which we know, okay? But these plasmids are not your typical plasmids. Usually they have been engineered to contain appropriate regulatory sequences for the transcription and translation. So usually the elements that is added is the promoters, which are stronger for the uptake and for the expression of the gene inserted into it, ribosome binding sites, and also for transcription terminators. For the eukaryotic protein and the expression vectors, usually eukaryotic proteins can be made in bacteria, and a lot of us, okay, even for me, okay, I would prefer it, my eukaryotic proteins, okay, to be expressed in a bacteria by inserting the DNA fragment into the expression vector from the prokaryote. So this is done, okay, because large amounts of this like protein can be purified from the transformed cells. But in certain cases, you also have to be very careful because the protein that could be used to treat patients, okay, with, for example, genetic disorders, sometimes, okay, could have a difference, okay, in terms of its nucleotide sequence. So you need to basically make sure that everything is in the correct order. So this is actually some examples, okay, for you to read on. Now, with everything, there is also disadvantages, and in this case, limitation of using the prokaryotic expression vectors. So prokaryotic cells, okay, usually are unable to produce functional proteins from eukaryotic genes. A lot of it has been done, even when all signals necessary, especially the signal protein for the gene expression, are present. Why? This is because eukaryotic proteins usually must be post-translationally modified, whereby your prokaryotic vector or expression vector do not have this capability, and they do have this capability, but not to basically post-translationally modified, okay, eukaryotic protein, because it's actually much more of a complex mechanism in eukaryote, which you will see in a few weeks, okay, from now. Okay, so I'll leave you with that. You can proceed on with the second video.